Okay guys, here we go with our next unit, structure and function of DNA. We talked about DNA briefly in unit 3 when we did organic molecules, and now we're just going to take it a little, we're going to delve a little deeper into the actual structure and then how DNA is used to make proteins and actually make our bodies work. So have your note packet out and ready. Here we go. Nucleotides. This is a review from back in the first marking period. We talked about nucleotides, okay? Nucleotides are what make up DNA, or any nucleic acid are made from nucleotides. So nucleotides are held together by strong covalent bonds. Between the sugars and phosphates. So the sugars and phosphates make up the sides. So when we talk about DNA, we talk about it being a double helix. So it's like a staircase that's been twisted around. The sides of the staircase, or the ladder, are held by, it's made up of alternating sugars and phosphates held together by very strong covalent bonds. So it's held together very tightly, okay? Nucleic acids are normally between several hundred and several thousand nucleotides long. So we talk about chromosomes. Chromosomes are made up of DNA. It's just one long DNA strand of thousands to hundreds of thousands to millions of nucleotides, all in order. 3 parts to the nucleotide. You have a phosphate group. That's here. Phosphate is PO4 minus. It has a slightly negative charge. So back in Unit 3, we drew a very simple diagram of a nucleotide that looked like this. Something like that, okay? So this would be the phosphate group, okay? And then in the middle, you have a pentose sugar. What that means is the sugar has 5 sides. There's one of 2 sugars. Either it's deoxyribose for DNA, or ribose sugar for RNA. So that's going to be one of the main differences between DNA and RNA that we're going to talk about later. But you have a 5-sided sugar, and then on one side you have that connected to the phosphate group, and on the other side you have it connected to a nitrogenous base. Okay, and the nitrogenous bases, again, review, there's 4 of them: A, T, G, and C. And just that each one of those bases, so you can see we call them nitrogenous bases because there's nitrogen here. Okay, and they have just a slightly different structure from one to the other. How did we get to figure out what DNA actually looks like? Well, it starts with Rosalind Franklin. She was using crystallized DNA. So she was actually crystallizing it and then taking x-rays of it to see the shape of it. So she was the first one to get kind of the shape of DNA, and her results when she took those x-rays, she was the one who determined that the DNA actually consists of 2 strands that are equidistant apart and they're in a helix form. So she was the one that actually first saw that double helix formation. However, James Watson and Francis Crick were working pretty much that same time to try to figure out that same thing. They were trying to figure out what DNA actually looks like. So they were putting together models. Some of them were like 3-stranded models. So they knew they had a basic idea of what DNA looked like, but they were having trouble actually putting the ideas together. And they were actually given Franklin's X-ray image without her knowledge. So they were credited with discovering the shape of DNA because they used Franklin's X-ray image to build their own 3-dimensional model of DNA, and it was the first one ever made. So Watson and Crick are the ones that are credited with discovering the shape of DNA, but the reason they were able to do that is because they used Franklin's X-ray image. They used her research actually without her permission and without her knowledge. So what they found out, 1953, they figured out we have a model of DNA, okay? So we have these 2 strands here, So all the way over to the right on your screen, these are the two sides of the DNA, and then in the middle, you have pairs of nitrogenous bases, okay? So we have a base pairing rule. I believe we discussed this back again in Unit 3. There will always be the same two bases that connect together in the middle. So the sides of the DNA, the sides of that ladder are always going to be alternating: sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, okay? All held together, covalently bond. Same thing over here: sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, okay? Those are the sides of the ladder. The rungs of the ladder, the parts that you actually would climb up, are two nitrogen bases bonded together with hydrogen bonds, and that's going to become important later on in this lecture, that the pairs are held together with hydrogen bonds, and that's why they're represented by dotted lines. The base pairing rule is that A always pairs with T, G always pairs with C. I like to remember this as all teachers go crazy. Eventually, all teachers go crazy for different reasons this year, because now, of course, we're going crazy because we're all cooped up inside. But that's my little mnemonic device to remember the base pairing rule. So A is always going to pair with T. So if there's an A here, there's going to be a T here. If there's a T here, there's going to be an A here. If there is a G here, there's going to be a C here. If there's a C here, there's going to be a G here. Those will always pair together. So because there's the A always pairs with T and G always pairs with C, another piece of evidence that was used to make this model is something called Chargaff's base pairing rule. So Chargaff was someone who was doing some experiments, and he was actually measuring the amounts of different nitrogenous bases in different species. So what he was finding was that the amount of A, or adenine, was always equal to the amount of thymine in a particular species or organism. And then the amount of cytosine is always equal to the amount of guanine, okay? So it doesn't matter, A doesn't have to equal C or G, but the amount of A is always going to equal T, because A and T are always connected together. And the amount of C is always going to equal the amount of G, because G and C are connected together. So using the results from Chargaff's experiment, and in addition to the X-ray of the crystallized DNA done by Rosalind Franklin, and then in addition, they found that there were two types of nitrogenous bases, okay? One type of nitrogenous base had a two-ring structure. So two rings. So remember how we're talking about we're talking about the actual base here, okay? And the only thing that separates the bases are the different, are how they're put together differently, the different structures. So certain types of these have a two-ring structure, and certain types of them have only a one-ring structure. And what was found was that something with a two-ring structure, one of those nitrogen bases that had a two-ring structure, was always bonded to one of the ones that had a one-ring structure, okay? It's the difference between, we call them purines and pyrimidines, okay? I'm not going to ask you to know which ones are purines and which ones are pyrimidines. I'm not really that it's not really that important, but you should know that one pyrimidine is always connected to one purine. So it's always one attached to the other. Adenine and thymine are connected by two hydrogen bonds in the middle, and cytosine and guanine are connected by three hydrogen bonds in the middle, which means that the bond between cytosine and guanine is just slightly stronger than the bond between adenine and thymine. Again, just a close-up. So you have all the way over to the right, that's kind of what DNA looks like when it's all coiled up, okay? So remember back to when we talked about genetics, and we talked about the structure of chromosomes. You have your double helix, and that twists up into your chromosomes, and it twists up and twists up and twists up until you get this like really strong coiled structure over to the right. This one on the left is kind of like the cartoony version of like, this is kind of what it looks like. So you have your sides of the twisted ladder. Those sides, again, are alternating sugar and phosphates on both sides. And then in the middle, you have your nitrogenous bases bonded together. Note the 2 hydrogen bonds between adenine and thymine, and the 3 hydrogen bonds between cytosine and guanine. All right, so DNA replication is a process that has to happen before cell division can occur. When we talked about the cell cycle and mitosis and meiosis, we talked indirectly about DNA replication because we discussed during the S phase of the cell cycle, S phase, the DNA has to be copied. Well, the DNA is copied in a process called DNA replication. Replication or replicate is just a fancy term of saying you're copying something, okay? So the steps for doing this, it's actually like, it sounds kind of simple. Now it's happening at all different places at all different times, so it actually becomes a little bit complex. But the simple portion, the simple steps is that the DNA helix is unwound, so that spiral staircase, that twisted staircase, becomes just a regular staircase, okay? It's no longer twisted up. And then the DNA is unzipped. What that means is that the hydrogen bonds in between the 2 nitrogenous bases are broken, okay? So I said before I was going to discuss why those hydrogen bonds are important. Those hydrogen bonds are important because they're super weak. They're very weak bonds, which means that you can very easily split them apart when you need to. Well, you have to in this particular situation. You have to split them in the process of DNA replication. So then an enzyme called DNA polymerase, remember your rules for naming enzymes, they always end in ASE, and then a lot of times they're named for what they do. A DNA polymer is a DNA molecule with many different pieces. So DNA polymerase is exactly what it sounds like. It's building a DNA molecule. Okay, so new DNA nucleotides, which are just kind of free floating around the cell, are added one by one to each side of the unzipped DNA. So think about those DNA sides. Once the hydrogen bonds in the middle are separated, imagine that they're both templates. Okay, so you can now read, the DNA polymerase can read. So let's say you have A, T, C, C, G, A, T, something like, so let's say you have something like this, okay? What the DNA polymerase, it's gonna read each letter. So the first letter it reads is A. Now what do you know? Well, A always has to pair with T. So the DNA polymerase is gonna find a free-floating nucleotide with a thymine and attach it, okay? And then it's going to read the T, and it's going to find a free-floating nucleotide with an adenine and attach it. And it's going to keep going like that, reading what the template is until all of the individual DNA molecules have a match. This is known as semi-conservative replication, and it's called that because conservative means you are conserving or saving something. Semi-conservative replication means that you are conserving parts or some of the DNA strand. So each new DNA strand is half of the original DNA, and then you have half of a brand new strand. So that's why we call it semi-conservative replication, because each new strand of DNA is half of what you had to begin with, and half is brand new. So this is kind of what it looks like here. So you have something called origins of replication. Like I said, it's a more complex process because it's going to happen at many different places on that DNA molecule all at the same time. So I told you that a DNA molecule can be thousands to hundreds of thousands of DNA to be in a row. It can be like thousands of them. So if you have replication happening at different points throughout that entire chromosome, it's going to happen faster. So you have your origins of replication, and that is where the DNA unwinds and splits. Okay, so now you have DNA unwinding, and notice how the original DNA strand here is blue. Okay, as the DNA polymerase is adding new nucleotides to that blue strand, now the new strand is yellow. Okay, so what you're going to end up with at the end is two identical strands of DNA. Half of it is blue, meaning half of it was the original strand of DNA, and half of it is yellow, meaning that's brand new.